So to begin today's webinar, I want to provide a small introduction to the GNA NOW Consortium. Then our project leader, Bacqué from Evotec, will go on more in details about the open call, what we're looking for, and what's in it for you guys, the interested candidates. And thereafter, I will explain a little bit more about the application process and end with a Q&A session. So just briefly, we are looking for a novel antibiotic program to progress to investigational new drug stage. And we think that this is a unique opportunity for people or organizations that have a gram-negative antibiotic or antibacterial. You will gain access to some of Europe's best laboratories and antibiotic drug research experts. And the urgency of the need of new antibiotics, I don't think I have to explain to this audience. So this is a little bit the agenda that I went over, and I am Kristina Orrling, who you see right now. You will soon see Erik, and I also mentioned Edwin Beyer at the very start, who is our legal officer, who might answer any questions that have that type of nature. So the GNA NOW project. We are a project under the Innovative Medicines Initiative umbrella, working on gram antibiotics, and it stands for New Gram Antibiotics Now. We're 11 partners, one of which is a big pharma, Evotec, who is a member of the European Federation of Pharmaceutical Industry and Associates. The total budget of the project is 31 million euros, where 2 million comes from IMI funding, so under the Horizon 2020 program. And the rest, 19 million euros, is an in-kind contribution from Evotec. It is part of IMI's umbrella, the AMR Accelerator, and we collaborate with other members and other projects in this accelerator. Currently, we're running 3 different programs, and we're going to enhance that with yet another one, your program. And they are enabled by 8 different platforms. It's a 6-year project. We started in July 2019, and we've been going on for almost 2 years now, and we will end at the end of June 2024. The aim of this project is to produce at least 1 phase 2 ready drug, 1 phase 1 ready candidate drug, and 1 preclinical candidate drug. The 11 partners are spread out in Europe. We have the North Bristol Trust and University of Liverpool in the UK, Erasmus University and Lygature in the Netherlands, Fraunhofer, and 2 Helmholtz institutes in Germany, Medical University of Vienna in Austria, and Evotec, Nosofarm, BioAster, and Inserm with their associate Université de Poitiers in France. Currently, we have 3 programs in pipeline. They are a little bit spread out in the value chain. For example, the NOSA 502 program is our most advanced project, which is close to candidate declaration. We're also working with a follow-up or related program, the NOSA 2G program, which is in is earlier in lead optimization, and the Karamisin program that is in lead candidate state. All these have a novel mode of action. This is very important also for later and for your programs. And they are targeting multiple gram-negative pathogens. But to de-risk the project goals, we have room for one more program, and that is the new program that might be yours. We have 8 enabling platforms, as I said. These consist of mode of action profiling, medicinal chemistry and design, microbiological efficacy and resistance in vitro and in vivo, PK/PD profiling, PK safety and ADME, CMC and fermentation. This is actually related to the current programs, which are peptidic in nature, and where biosynthesis has been explored, and clinical studies and modeling. It works as we have the governance of the programs are secluded for the particular program. They are part of the work packages, that's the WP. And we have a steering committee for each program. And they are interacting with a selected number of the enabling platforms and enhanced by external experts who are also participating in the respective steering committee meetings. In addition, we have a scientific advisory board with external experts who are giving input on the general questions and leadership of the consortium. Work package 1 here is the incubator management. That's me, Rick, and Karin, basically, and Sophie Lagrange, also from Evotec. And this concerns more the governance and making sure that we're reaching the project aims. So just briefly for this open call, it was launched on 5th of May. A very important deadline is the 18th of June, which is the deadline for the submission of a so-called expression of interest. This is a limited sized document for the first selection. It will be reviewed by our review committee, consisting of 4 external experts and 3 GNA NOW members. They will score and rank the different expressions of interest, and the top 3 will be invited to submit a full program dossier where a confidentiality disclosure agreement will be needed, so we can exchange also confidential data. And the deadline for the submission of a full program dossier is 25th of August. So now I would like to give the word to Eric to explain more what we're looking for and what's in it for you. Please, Eric.
Yes, thanks, Kristina. Good afternoon to everyone. So I'm Eric Bacqué from Evotec. I'm the project lead for GNA NOW Consortium. Next one, please. So first slide is about what can you expect from GNA NOW, what can you look for in this consortium. So we are looking for a lead to candidate program or a development candidate that is under or ready for preclinical development. This is the key point. So we are looking for advanced asset, I would say. In terms of profiles, so this asset should be TPP consistent spectrum. They should have acceptable resistance risk. satisfactory ADME PK properties, demonstrated in vivo efficacy, established IP position, and suitable safety profile. The asset should address severe hospital infections with a preference on ADVAP, but also CUTI, or urinary tract infection, intra-abdominal infection, and bloodstream infection caused by gram-negative. This is really the key point. And so this is covering Enterobacteriaceae, Pseudomonas, and Acinetobacter. We have a preference for broad-spectrum TPPs, but we can allow single pathogens. But in this case, it should be non-fermenters TPP like Acinetobacter or Pseudomonas only compounds. Another key point for this asset is that we are looking for novel mode of action. So meaning no cross-resistance with marketed classes of antibacterials. So this doesn't really necessarily mean that we are looking for new targets, but this can be a non or underexplored target that can be modulated by a new way, for instance, by binding to a new site in the protein, for instance. We are looking for directly acting antibacterial effects or potentiators, but we are excluding antivirulence compound or any asset that would not show MICs. Next one, please. So in terms of modalities, we are looking for small molecules with a large definition, so it can be also natural products, small peptides, but non-biologics, so no antibodies, for instance, or no lysins. We are looking for potential for intravenous administration. This is really mandatory, and this is linked to the TPP we are looking for. Of course, oral administration is not required, but could be a very nice added characteristic or property of the asset. And this is really needed, we think, in the case of the Enterobacteriaceae only TPP, so meaning for the CUTI-based TPP only. In terms of technical feasibility, the program that is proposed should match with available GNA NOW resources, capabilities, and expertise. And they should have the potential to benefit from initial discovery and development expertise and capabilities that are brought by Evotec. Also important is that the priority will be given to organizations eligible to IMI provision for beneficiaries or FPR companies. Next one. So what's in for you? In terms of resources, we are talking of several millions of euros, and this the exact amount has to be defined according to the project needs during the preparation phase when the final candidate has been selected. Of course, you will have, as mentioned by Kristina, access to some of the best European laboratories and experts in antibacterial drug discovery today. You will have also access to all the components that are needed to progress your compound to phase 1 ready stage. This is an important point. There will be a partnering contract with Evotec. This is mandatory. This is a concept of the consortium. So we are a pharmaceutical company with the experience, the expertise, the resources, the platforms that are able to bring a drug to clinical development stage. You will keep, sorry, previous one. So you will continue the control on the development of your project as a member of the compound steering committee that are built for each program. You have also the option to contribute to a GNA NOW according to 2 modes. First one is the so-called contributing third party, or as a full member of the consortium. We will come back to this. So next one. In terms of evaluation, so there is a review committee that are evaluating the OI and then the confidential presentation, and there is a score system from 1 to 5 for various topics. So the first one is scope, innovation, and novelty. And so this is in line with the minimal criteria defined for the call. We also take into account indication, TPP, development stage, competitor analysis, etc. Next topic is about excellence. So about the data that are available for the compound on the series. This is also defined with respect to the lead and preclinical candidate criteria that I will present later. In terms of feasibility, the third point, so how well the program matches the GNA NOW resources, expertise, and timelines. What can the compound owner contribute with? And finally, implementation. How mature and realistic are the project plans and timelines? So, in other words, can the project really fulfill its objective during the lifetime of the consortium? Next one. So this is the lead criteria, at least the main criteria. I would say there are more in the document that you can access to. So in terms of mid-90s, so they should be below or equal to 4 microgram per ml for the targeted Enterobacteriaceae and below 8 microgram per ml for the targeted non-fermenters. The specificity of and the on-target antibacterial effect should be demonstrated. So this shouldn't be the MIC shouldn't be due to some membrane perturbation and specific membrane perturbation, for instance. In terms of resistance, you should demonstrate that the risk is manageable for the key TPP pathogens. In terms of profiling, FISChem, ADME, ETOX properties, they should be documented for the lead compound and their analogs, and you should avoid the accumulation of too many alerts. So 3, 4 is already good enough to expect to have a rapid optimization process. PK property in mice should be compatible with TPP indication and routes of administration. In vivo efficacy should be demonstrated. This is really mandatory, and it should be in the TPP relevant infection models with concomitant tolerability. In terms of chemistry, the synthesis should be tractable and scalable, allowing further optimization of the series with rapid optimization. You should have at this stage an acceptable IP position and lead to candidate optimization strategy, flow chart should be already defined, taking into account the liability of the series. One important point is that the proposed lead criteria that I've just listed are just indicative, so meaning that the lead compound, your lead compound may not satisfy exactly all the listed quantitative criteria, or a few properties may not be properly documented. But as always, what will be evaluated, this is a complete picture, and so the lead dossier is evaluated as a whole and with respect to the distance to the ideal lead profile. So same exercise for the development candidate criteria. So the mid-90s are slightly better than for the lead, so 2 microgram per ml for the Enterobacteriaceae and 4 for the targeted non-fermenters. Risk should be still manageable on TPP pathogens, and the mechanism of resistance should be documented. You shouldn't have any major in vitro and met warnings. In vitro efficacy should be demonstrated here for multiple strains and multiple infection models, representative of the targeted clinical indications. PK available for several species, so not only rodents, but also non-rodents, and by the intended routes of administration, of course. You should have acceptable in vivo tox profile and safety margins in rats. Efficacy driver should be determined based on the first PK/PD studies. You should have a reasonable projected active dose in human, at least a preliminary estimation. You should have also demonstrated capacity to deliver large batches of the candidate, so typically 100 grams of the compound, and the IP position should be compatible with future FTO. And again, you don't need to tick all the boxes, but at least to be as close to this ideal portrait. Next one. So this is about the selection criteria in terms of feasibility and implementation. So feasibility, in terms of resources, your contribution, including funding and expected support from GNA NOW should be provided. Additional resources and expertise that are assumed to be not supported by GNA NOW Consortium should be included and should be, of course, ranked as subcontracting. Contribution from GNA NOW partners are expected to be the following: so mechanism of action and the target elucidations. This is for LMODs and bioaster. In vitro, in vivo microbiology, so this is NBT and INSERM. In vivo microbiology and PK allometric scaling human doses prediction at Erasmus and University of Liverpool. Other platform like medicinal and computational chemistry, social biology, protein production, ADME, PhysChem, CMC, in vitro, in vivo toxicology, and pharmaceutical sciences can be expected from Evotec. The budget, you should provide an estimated budget for the GNA NOW activities in line with the consortium activities. So we should have an idea of what could be the cost. So is it compatible with the budget that are left? In terms of implementation, the plan will be discussed in collaboration with the partners, with Evotec, and with the compound owner, of course, during the final stages of the preparation before entry into the consortium. This document will include action plan to reach the next milestone, the timelines, even if indicative at this stage, essay cascade, so the flow chart, the task, deliverable, and responsibilities as well. So this is for the implementation part that should be part of the dossier. Next one, please. So this is up to you, I guess, Kristina.
Yeah, so the expression of interest template, it's available on the web, so on the Open Call site. This can be mailed out to you after this meeting, so you can have it at hand directly, and also the document itself. The outline is basically questions that are reflecting the selection criteria that Eric was spelling out. The first question is really of administration nature. We need to know who is applying and who owns this compound, or who has licensed the compound that you're submitting. Then a short history of the program, and then the target product profile. There's a table provided to fill in. And then some data on the target, chemistry, maybe CMC, especially for a preclinical candidate. The microbiological profile, PhysChem properties, PKPD properties, the in vivo efficacy and toxicity data. PKPD studies may not be applicable for a lead optimization program. And then the known liabilities and risks. I think this is, it's important that you do expose yourself here and be open with it, because it will make it possible for us to judge whether this is something that we can tackle within the GNA NOW project. And then the foreseen development pathway. What do you judge need to be done, and indicative timelines. So please remember to not include any confidential information, to put everything down on maximum 5 pages. So we want to have a brief description of these things. Think about also adjusting your answers to the stage of your program. Indicate also whether you have relevant data that you cannot include for the sake of space or confidentiality, but that you may be able to include in the dossier, would you be selected. And please contact the GNA NOW office in case you have any questions. So I will just briefly show you what the dossier template is. It's in a PowerPoint presentation format, and it contains more or less the same questions as the expression of interest, but more in detail. For example, intellectual property, we want to have a little bit more information on that to be able to judge. So, and the same thing, for example, in section 11, we want to see CNC, what has been done for scaling up, etc. But here we will be much more in closer contact with the candidates that have been selected for submitting a dossier. So your proposals, the expression of interests, will be submitted to the email address gnanow@lygature.org. And if you have any questions up front, I know some of you have already contacted us, please involve us. And please involve us when preparing the expression of interest. We might be able to help. We cannot tell you how the reviewers will look at your proposal, but at least help you prepare it according to our expectations. I did discuss the timelines already. So the important one is the deadline for submission, 18th of June. Then it will be reviewed and scored and ranked. And by 7th of July, we will contact the 3 proposals or the 3 compound owners that have submitted the highest 3 ranked proposals. And to start preparing the dossier, and in parallel, we will establish a 2-sided confidentiality disclosure agreement with your organization. So by the deadline of the 25th of August, that you can disclose confidential information, and we as a consortium can also share confidential information such as our consortium agreement with you. And you will have the result from the evaluation of the dossiers on the 15th of September. So by which time the top ranked proposal will be invited to sign contracts first with Evotec and then with the GNA NOW Consortium. After that, we also need to define how the new program will look like. And this has to be done together with the GNA NOW partners, who will do what work and when and by which time. And we will establish a core team. These are the teams that are leading, that doing the daily operations of the programs, define a steering committee for this particular program, and invite external experts to give advice. We also need to define the go and no-go criteria. So for the decision milestones that define whether it will get continued support from IMI and the GNA NOW Consortium, or whether we need to suspend this project. And we also need to submit a revised description of action, which is part of the grant agreement with our funders, the Innovative Medicines Initiative. And this explains it in text. I won't go over again. Some important parts is the IP and access rights, which is defined in this slide. And I think this is something that is important for your organization to consider also upon the submission of expression of interest. So this can be further read, and we can explain this further also upon questions. These are relatively common rights and obligations in European collaborative projects. We also discussed earlier that you can join the consortium in two different ways, either as a beneficiary or as a contributing third party. You should be aware of that it is only organizations that either belong to FPA, so the European Federation of Pharmaceutical Industry and Associates, or the ones qualifying for the IMI2 joint undertaking funding that can join the consortium as beneficiaries, and that is as full members of the consortium. In principle, any legal entity can join us as a contributing third party. Beneficiaries, they will join the full, they need to agree upon and sign the full consortium and grant agreements, while contributing third parties have a limited agreement to sign, which is what we call the contributing third party agreement. The resources that we can give is the GNA NOW in-kind activities to both types of compound owners, but a beneficiary might have the chance to also receive monetary funding from the IMI. And then we have the governance. I mean, what can you influence? Both can influence your own program, but the contributing third parties are not part of the bigger consortium in that sense that they cannot influence the bigger project decisions. Also only have responsibility for their own program and not for the project as a whole. And both type of compound owners need to sign the bilateral agreement with Evotec. So again, I repeat, submit your expression of interest before 18th of June to get the chance to join us in our endeavor for finding a new antibiotic. So with this, I want to conclude this part of the webinar and open up for questions and answers. And by this, the recording will also stop.